If you could share your slide as I introduce you, sir. So Dr. Patrick McDermott is the director of the National Antimicrobial Resistance for Monitoring System, or NARMS, at the FDA Center for Veterinary Medicine Office of Research. He is also the director of Division of Animal and Food Microbiology. NARMS was established in 1996 as an interagency collaborative effort of the FDA, USDA, CDC that tracks antibiotic resistance in bacteria from retail meats, food-producing animals, human clinical cases, and illness. Dr. McDermott began his career with a BS in zoology in Arkansas, and he continued there with master's as his PhD in microbiology and immunology at the University of Arkansas Medical Center. He has published more than 150 peer-reviewed journals, more than 120 since 2000, 25 reviews, multiple book chapters, as well as book editing. He's one person who said he has so many abstracts and conferences, he actually has stopped listing them since 2005. As you can see, Dr. McDermott has worked over 25 years in the field of antimicrobial resistance. He's a fellow of the American Academy of Microbiology and recipient of the FDA Francis Kelsey Award for Excellence and Courage in Protecting the Public Health. And it's with great pleasure, Dr. McDermott, if you could begin. Thank you.
Thank you very much. Thank you for the introduction and the invitation. I can't quite tell what you're seeing, so I'm going to let me know if this appears correctly to you all.
Yes, and if you, we can see your screen, sir.
Okay, thank you very much. Well, thank you again for the invitation. I appreciate the opportunity to talk about what we're trying to do in our national AMR surveillance to, let's say, transform the program into a framework that is consistent with the principles of One Health. And that'd be the theme of my presentation today. We began this process, well, first of all,
McDermott, if you could share the full screen.
Yeah, see, I don't know why it won't do that because I see the full screen on my computer, so maybe I need to share a different screen. Let me try that.
Pat, just a slideshow. I think we can see the slides, just the bottom, the full.
Oh, yeah, see, they're all grayed out on my computer. Let me try this way. I think I must be sharing the wrong screen. Is that any different?
Perfect.
Okay, thank you. It's okay. So you can see the full screen.
Yes.
Okay, thank you. So yes, back to the topic of the presentation today is an outline of our attempts to make NARMS into a program that's consistent with the principles of One Health. The program, as was mentioned, NARMS has been around since 1996, 25 years now. And it is the U.S. National Program for Tracking Antibiotic Resistance in the food supply, mainly in the food animal and derived meat supply. And it's been designed along those lines for those past 25 years, where it's looked at resistance in sort of the farm to fork continuum. And we had NARMS reviewed in 2017 by a subcommittee of an FDA science board, which we typically call on or routinely call on to evaluate programs at FDA. Dr. Barb Kowalczyk, who is part of this program, was on that review board. They looked at the status of the program and made recommendations on how it can make progress and become better in the future. And this was done, of course, based on a One Health model of monitoring, which we all know is now a rather consensus approach to addressing infectious diseases and advancing infectious disease science in general. We took the recommendations from the Science Board Committee, we combined them with our own thinking on best way forward and held a meeting in May of 2019 and developed a new 5-year strategic plan for NARMS. And we had a rather fruitful brainstorming session and reduced some 130 or so recommendations down to a set of 4 major goals and 16 objectives. And the theme is both One Health and genomics, and the two actually go together quite well. So The recommendations from the Science Board in transforming it into a true One Health program was, we know the definition of One Health includes human and animal health in terms in reference or in the context of a healthy environment, let's say. And so it was one obvious step was to add animal pathogens and make animal health a principal component of the surveillance system. There was recommendations to develop an environmental surveillance piece. This is probably the most complicated recommendation that came out of the review and one of the most complicated things to implement. The environment's a big place. There's different sampling points. They're all going to provide different types of information. So we've really struggled with this, and I'll show you what we've decided would be a logical place to start in terms of environmental component to NARMS. There were recommendations to add other food commodities, test more bacterial species, if possible, get on the farm and collect samples on the farm. Now, I will say in the U.S. that's a very difficult prospect and something that I don't know how we'll be able to do it efficiently and affordably in a representative way. So I consider that perhaps a bridge too far, but others on the call may have some thoughts on that as well. There were recommendations to continue to exploit next generation DNA sequencing technologies and develop methods of microbiome surveillance by getting important metadata or more informational metadata that would help in other food safety priorities beyond simply AMR surveillance. And I will say that with genomics, that's become almost an automatic thing since these data sets are publicly available. It's present in a common language, namely DNA nucleotide sequences, and people can apply their different analytics to the data set. So we'll come on to that, but sharing the data with everyone in near real time is one of our objectives. There was a recommendation to better exploit whole genome sequencing to get down to hidden outbreaks in sporadic cases and make better informed decisions on clusters and that perhaps were overlooked before. And then a signal to broaden our collaboration both domestically and internationally with partners to really come up with some sort of agreement on what the best practices are in One Health AMR surveillance. None of these recommendations came with a handbook and definitions. I'm getting two different screens, but perhaps I'll carry on. So let me see. I'm sorry, I'm having a problem seeing the right slide on my screen. Okay, there we go. So this slide attempts to sort of just provide a characterization of what the old and new look like. So in the past, right, we were always looking at zoonotic foodborne bacteria, looking at the same pathogens in food-producing animals in their derived retail meats and in human clinical cases. And this was defined by WHO as integrated surveillance. That is the use of coordinated sampling and testing of the same bacteria from the 3 different domains and the subsequent evaluation of the resistance trends using harmonized methods. So kind of a linear approach. But now with One Health, it's beyond zoonotic foodborne in a sense, and it's to look at these interacting environments and ecosystems of the human, animal, and plant environments and doing it in a new multidisciplinary collaborative way, working at local, regional, national, global levels with the goal of achieving optimal health outcomes and recognizing these interconnectedness of the environment and human and animal health. So that's the CDC definition. Perhaps you could say the definition of One Health AMR is the first one, and we simply add in there, and the environment and animal pathogens. And maybe that is the right definition of One Health AMR surveillance, but that's what we're trying to grapple with in this new strategic plan that we've just recently published. And perhaps some of you were on our public meeting a few weeks ago where we went over the strategic plan. But I thought I'd present sort of our plans and our progress within the context of that plan. And with One Health in mind as the model, sampling, of course, is the key first thing to consider. And sampling within a One Health framework requires a new partnerships to collect new data sets that help us get animal pathogen information and define something that is a reasonable environmental testing scheme. So here are the objectives we've put out for sampling within a One Health framework. First, to enhance and maintain routine resistance monitoring and pathogens from food-producing animals and companion animals. And we've made some progress in that arena already. The second objective is the environmental piece. So to implement representative monitoring, including surface waters, to establish baseline AMR data in aquatic ecosystems. So that's what we've decided was a logical place to start in terms of implementing an environmental monitoring component. It's a difficult thing. There's a lot of good reasons to criticize any approach, but I'll share with you why we think this might be at least a good place to start. Third objective is, if we're looking at human food and resistance in humans, it seemed like a logical parallel to look at animal food and resistance in animals. And so we at least have let's say, a long-term goal to have periodic examination of pathogens that might be present in animal feed and pet food is one way of addressing the animal health component. Objective 4, so this is where we get beyond the traditional testing that's been done in NARMS, which is looking at terrestrial animal species in chickens, turkeys, pigs, and cattle, to look at other sources of protein that are raised with antibiotics. And a big one, of course, is seafood. Seafood is mainly an imported product, so this is a little bit outside the historical boundaries of NARMS, because the FDA, anyway, uses these data in NARMS to help in its regulatory activities, and we don't regulate antibiotic use in imported products, but we do have an interest in exposure to potential resistance through any source of food that is raised with antibiotics. So seafood is added to the list, and we completed a pilot study looking at shrimp, tilapia, and salmon last year, and we've started full-time testing of seafood products this year, full-time with, of course, interruptions from COVID. The other piece in here is to look at in any domain where animals are reared with approved antibiotics, and so this includes other food-producing animal species, so-called minor species. So we've started looking at some of that with our partners at USDA, where at the slaughterhouses they're now also looking at veal and lamb and goat and catfish. So all raised with approved antibiotics. But the environment and the depth of sampling and the importance of both plants and animal health definition. We also are interested in produce and potentially wildlife. There's questions about how some of these resistant enteric pathogens spread so quickly when they appear, and we've seen that in many cases with Salmonella DT104, with Salmonella infantis more recently. And how do they move so quickly up and down the hemisphere, for example? And maybe looking at migratory wildlife is at least an interesting academic question that might help us answer some of these risk gaps. And then lastly, if the first three objectives are a broadening of the scope of NARMS, the last two are a broadening of the depth of testing. And so we're trying to look at other organisms as well, beyond the classical ones. And so if we look at what the strategic plan is leading us to in terms of trying to implement best practices in One Health AMR surveillance, as well as deepening our sampling to get at more types of commodities raised with antibiotics. The red boxes here show the changes that we've made recently to pursue these objectives. So you can see that if you look across the top, the Centers for Disease Control is looking at human isolates of enteric illness. At FDA, we're in 23 states looking at retail meats on a monthly basis. USDA is looking at food-producing animals and has a very robust nationally representative randomized sampling scheme to get these bacteria that we test in common out of food animal intestinal contents from slaughter plants around the country. And then if you look in the next row, it shows what bacteria or what samples are being tested, and with the humans and persons, and with the retail meats, like I mentioned, we've looked at terrestrial animals and the products in the past, and now seafood, and correspondingly the food-producing animal piece is looking at the terrestrial food animals, but also new ones, including veal, lamb, goat, and catfish, as I mentioned. And so on the animal pathogen side, so this is new. This is something we started doing in 2017, and that's in partnership with FDA's Veterinary Laboratory Investigation and Response Network, VetLARN, and also USDA's National Animal Health Lab Network. And they're essentially harvesting information from the network of vet diagnostic labs around the country and looking at pathogens important to animal health. Now, I don't have the environmental piece in here yet because we haven't really gotten that off the ground entirely, and it's still in the development phases. But I think it's fair to add NCBI and NIH, National Center for Biotech Information, to the NARMS franchise, if you will, since all the submitted genomics and sequencing get deposited there. And also, we've worked very closely with them to develop develop tools to screen all the genomes for known antimicrobial resistance genes. So on the animal pathogen side, this is just a basic outline of how it's working with the VetLearn partnership. And so they've got 45 laboratories around the country, with one in Canada, and they're getting the first 4 isolates, I believe, of the pathogens listed here, testing it for susceptibility using the methods that the labs rely on, but then making sure that we standardize the output in terms of reporting resistance rates. And then again, and then the bottom right, you can see a table of the progress we've made so far. And it began in 2017 with about 1,700 total isolates of salmonella from all animal hosts, and then staph pseudointermedius and E. coli from dogs, the number of susceptibility tests, and then the whole genome sequencing at the bottom. So these data, as in the rest of NARMS, are being uploaded to the national databases for sequences. And so you can look at our reports and see some of the preliminary data on that. We continue to expand the animal pathogen work, and we're very happy to partner with anyone and to get feedback from anyone on how we're doing and how we can do this better. So on to the other One Health sampling piece, it's the environment. And we took our cues from a meeting of the UK and the US, the CDC in the UK, let's say, to look at antimicrobial resistance in the environment. And it's really a very good report. It's very logical. It draws clear boundaries. It explains data gaps. And it talks about the major. It's essentially an attempt to incorporate into surveillance, or at least into our general public health understanding, what the impact on the environment is of runoff of AMR from built human and agricultural environments. That's probably a good way to summarize it. And the report points to, say, hospital effluents or human waste through the sewage system, the application of animal manure to fields or runoff from feeding operations, but also aquaculture, where antibiotics are put directly into water ecosystems, and runoff from the crop use of antimicrobials were all listed as potential inputs into the environmental AMR burden. And so we took that and combined it with our own discussions and looked at surface waters as probably the best place as a confluence point of all these built environments. And so we're working right now on pilot studies to look at the geospatial distribution of resistance. And the idea is to build this into a risk model, to look at sources and selective pressures for amplification transmission, and then define standardized sampling. And we're working with the Environmental Protection Agency on this because they have such good data on the watersheds around the country, including both sewer overflows, which would put untreated sewage into the environmental waters, animal manure application and production. There have been calculations. There's a paper here listed on the top right to show how much of this water actually is recycled that we actually consume again after it's been treated from runoff through downstream intakes, etc. You can look at that study and get some information on that. But we reuse and re-expose ourselves to wastewater eventually. And then lastly, the thing that I find most helpful in trying to put this environmental piece together, and this is the bottom right map here, is that the EPA has categorized rivers and streams according to their impact based on some physical and biochemical and biological indicators, such as pH and the biosphere within the streams, riparian testing, sediment testing, chemical levels, temperature, pH, etc. And they can come up with a scale of impact based on these measurements. And they define them as like high impacted watersheds, which are the red dots, intermediately impacted are the yellow dots, if you can see them, and then blue are sort of the reference or pristine waters. And so this makes it possible to combine data on AMR with data on impact of the environment from human activity, let's say. So where this stands right now is they're in the process of developing a national scale quantitative assessment of AMR within these surface watersheds, working on standardizing measures to monitor trends, input models for risk, help quantify drivers and occurrence of selection pressures for the resistance that is seen, and then maybe critical control points to do something about it, to take action based on a risk model. So that's what we're trying to do in terms of sampling, in terms of exploiting genomics. We have as goal to employ advanced technologies, and that's really talking about next generation DNA sequencing and computer machine learning and artificial intelligence to really dig deep into these detailed data sets that genomics provides. And so we outline here to do just that, to apply machine learning, to use metagenomics, to employ long-read DNA sequencing methods to get closed genomes. So we have a reference of coding regions for known AMR genes and the DNA elements that motivate their spread between bacteria. And then lastly, because we're getting so much information, and as we all know from genomics, we're seeing new insights into things that travel together in resistant pathogens. And so research to understand adaptive microbial features that might contribute to or affect the spread and persistence of resistance is part of this. And it's mainly a bioinformatics exercise. So in NARMS to date, we've got, this just summarizes that about 52 isolates where we've done antimicrobial susceptibility testing and whole genome sequencing. So as you can imagine, we're looking at salmonella from 14 sources, resistance to 15 antimicrobials, and then 3 or 4 million base pair nucleotides of data on the isolate itself. So it's a big, complex data set, and we're trying to take full advantage of it to understand the movement of resistance in the One Health context and present the data in a way that's accessible to a broad base of stakeholders who have different levels of interest and understanding. But we're pushing as hard as we can to see how these genomic tools can work in AMR surveillance. And so with these 52,000 genomes where we have susceptibility data, we've shown that you can, if you look for known resistance determinants in the genome, you can predict MICs at the resistant breakpoint with a very high degree of reliability, over 90% overall, or 95% overall, and around 90% for some of the weaker ones. But you can accurately predict resistance from the genome. So this really, and this shows our papers where we've looked at it in the NARMS organisms. Well, that has huge, that's profound implications. That means we can do essentially genomic susceptibility testing on any genome submitted to public databases. And so immediately when you think in terms of One Health, we've got a tool that allows One Health AMR investigations to be applied to just DNA sequence data, good DNA sequence data, and not the need for the in vitro susceptibility testing, or at least we could use the data without that traditional method of predicting resistance. And we took it a step further. You know, there's one concern as well, but how do you know what the MIC is? And so we did a collaboration with Argonne National Labs at the University of Chicago, and we took 5,000 at the time of our genomes, and without any, we trained, this is a little bit beyond my understanding, but we trained using known susceptibility patterns to 16 antibiotics, and then used a K-mer-based approach to form a matrix and show how well can we predict a given MIC across the range of testing that we do. And if it turns out you can actually predict any MIC with that great degree of confidence, 95 to 96% within a single two-fold serial dilution, which is the standard error on just phenotypic testing itself. So you can predict them with no a priori information about the underlying content, and you can do it with as few as 500 genomes. So the method works really well for predicting resistance and predicting MIC, perhaps. We need to do more work there. So that led us to a new way of interpreting the data, right? If we're not doing phenotyping, how are we going to predict the genotyping data? And even the phenotyping data is used differently in Europe, at UCAS versus the US. And so they have different breakpoints for different drugs, say for salmonella in this case. And Greg Tyson, our group, said, well, let's look at the highest MIC that don't have any known genomic or genetic elements, and came up with this concept of genomic cutoff values. And it shows that in the red font that maybe some of these discrepancies between the US and the EU on interpretive criteria can be resolved now with genomics. I mentioned the incorporation of closed genomes for getting detailed catalogs of resistance genes, and we're up to 150 or 200, I think now, closed salmonella genomes from different serotypes and different resistance patterns, and we're getting complete detailed analysis on the sequences of the plasmids of these, and we'll put these up on the web for other people to refer to. So all of this really is propitious. This combination of sequencing and One Health as a framework is really come together at the right time. And there's a study I've used this for many years in my presentations, but it's still such a good example of how good sequencing is as a method for One Health AMR monitoring. We did a sequencing study back in 2007 when it was still very expensive to sequence genomes, and we looked at paired serotypes of salmonella from susceptible and resistant patterns and sequenced them. And this is one example of a salmonella Newport that had this INK-C plasmid that is just teams with resistances, and you can see the different resistance genes here. We're interested in expanded spectrum beta-lactamases and Blossium Y2, and we found it on a plasmid with resistance to aminoglycosides, tetracycline, chlorophenol, sulfonamides, and mercury as part of TN21. But also on the bottom right, you can see resistance genes to acetylpyridinium chloride, the SUG-E gene. Well, that's a compound used in processing plants to treat carcasses in U.S. Food animal processing. So that was quite interesting, and it travels in a copy right along with the CMY gene, which is the one of critical importance for treating these infections. And then we found the same plasmid. It had been seen before in a child in Madagascar 10 years before who had plague, and we found out it was pretty common in the U.S. retail meat supply, and it had reached a species of Yersinia ruckeri that's a major cause of septicemia in fish. And so I think this is just such a good example of the tool of sequencing to address One Health AMR. We saw a single plasmid that moved quickly around the globe in different genera, gained resistance to many major drug classes, acquired traits to thwart treatments in the processing plant, probably affects the resistance patterns and strains coming out of that processing plant. In addition, it's making red mouth disease in fish more difficult to treat and plague in people more difficult to treat. So it's an old example, a good example of the One Health nature of AMR and the value of sequencing to reveal these traits. And so there's a Genomics and Food Safety Coalition in the U.S. government that's adding to antibiotic resistance other types of food safety relevant traits and other traits that we can sort of paint over our strain databases to give this detailed level of information according to people's interests, whether it's heavy metal resistance. And I've underlined the ones that are being pursued right now. Virulence genes are now present at the NCBI database, biocide resistance, heavy metals, and MLST types, et cetera. So there's a lot of different relevant data sets or data that can be inferred from the genome and help in addressing food safety issues and AMR issues. We're also on the metagenomic side, using metagenomics to try to see what the different resistome patterns look like in different food animal species. And so we took several years of the USDA sampling at slaughter, 25,000 sequence samples we received. We reduced them down to DNA and are sequencing a subset of them and getting these sort of baseline levels of resistance in the animals. And I don't have time to go into the value of this. Hopefully it's obvious, but this is a good way to get to, say, outbreaks where you don't have a food source and to the consequences of drug use in different species. The other way we're using metagenomes is on the waterworks. And so we did a pilot study in the northeastern United States with EPA and looking at these more and less disturbed water systems I described that they'd categorized, and then applying metagenomics to these samples to see what the resistome looks like in surface waters. And this shows, just looking at the taxonomic structure of the less impacted, the green circle R, and the more impacted surface waters, T. And you can see there's dots in the middle there that show you the number of bacterial families that are shared between reference and impacted waterways, and then the unique bacterial families to each. So you can see in the reference strains there's a single family unique to, let's say, the pristine waters, but in the disturbed waters it's got a great deal of diverse microbial diversity, as you might expect. And then using the SQL data that I just described, we've got some comparisons showing how the overlap between the different major food animal species and what's seen in the waterways of impacted and reference waters, how that sort of plays out. And you can sort of see something like a weather map developing from this. And when We go to look at the resistances. Again, if you look at the big red T, the impacted waterways, a great diversity of resistance. Some shared between them, maybe as a dozen or two shared between them. Very few unique to the reference waterways. So it makes this is maybe what you would expect if the waterways are impacted by human and ag activity, that there would be more diversity of organisms and resistances. And so here we combine or did the same sort of analysis as before based on the resistance we've seen in the metagenomes of animals with what we've seen in water. So this is a very early studies with a fairly small number of samples, but this is our thinking on the approach here. I got maybe just a minute. I'll try to go quickly through these last bits. The last two is one is the third goal is improved data sharing and collaboration. One thing that we're going to release in about a month is predictive resistance. So this is going to be just resistance based on the DNA sequence. And, we've had a hard time getting this information out to people who can use it. And sequencing allows us to get around that. We're going to start putting data up on a weekly basis eventually, and if not a daily basis, based on predictive resistance. And so instead of waiting for our 2018 report to come out, you'll see our 2020 data as of last week. And this graph on the right shows the blue box on the right will be predictive resistance. And then that should make the information available. Our goal is within a month of collecting the sample, the sequence data will be in the public domain. The other tool we're working on for data sharing is Ohm Tracker. This is essentially taking resistance genes and using them, regardless of the source, and using them as a tool for people to look at resistance. And it's really harvesting the information from NCBI, and you can, and then putting it into tools that make the data accessible for others. And so what this shows is that you can go into Ohm Tracker for Salmonella, and you can start with the resistance gene you're interested in. So we just decided to reverse sort of the process and for people who are doing resistance work, and then pick that gene, get a list at the bottom. There's a list of all the strains that have it, and then find the strain you're interested in, and it goes right to NCBI's pathogen detection portal and puts that isolate on the phylogenetic tree, and you can see, whether it's associated with outbreak strains or other strains in the database. And yet we're also trying to put out there, although the metadata need cleaning to make that data accessible across all of the ontologies that are part of the metadata at NCBI. And so you can see here the number of isolates for these different sources of the different metadata associated with the genome submitted to NCBI. So we're hoping that'll be a useful tool for people who want a more global view of it. And then lastly, goal number 4, conduct research. This is, I guess you can summarize the last goal is to sort of signal our willingness to work with anyone who has variables that NARM surveillance might be able to detect if they implement any type of intervention. So if vaccines come out and maybe replace antimicrobial uses, well, we'll be able to detect things like that in the surveillance. Can we better understand risk factors for resistance and resistance in animal pathogens as well? We can't figure everything out by surveillance, I guess is another way to look at it. So this is a familiar graph. This shows how we've become, or how we've expanded with this new strategic plan. So if you look at the green boxes and circles, that's what NARMS was in the past. With the new strategic plan, the blue, you can see how we're getting more inclusive of, let's say, the ecosystem as a whole, moving to look at imports, to look at animals, to look at animal feeds, to look at surface waters and rivers and streams. Perhaps aquaculture, I didn't mention that, but we're looking at aquaculture pathogens as well. And perhaps wildlife and sewage might be part of what we do in terms of environmental testing in the future. So we're starting to expand and enhance the scope of the work. So let me summarize. Best practices in One Health national AMR surveillance are not fully defined. I think we're doing what we can to contribute to that conversation globally by the things I've outlined for you this morning. Incorporating animal data pathogens, obvious, but there are many animal pathogens, either they're not tested for susceptibility or the data aren't available. And, it's very different in the animal world. You don't have insurance companies paying for tests like this. So many times they're simply not done and the data aren't there. The environmental testing has begun with the pilot projects I described. It does lend itself to risk analysis. We think it's a logical way to begin if and we're open to feedback and course adjustments as we learn more and more there. We're expanding the scope to more animals raised with antibiotics and exploiting next-gen sequencing technologies as best we can. I think I'm about on time. I hope that was helpful to you. I would just end by saying that, repeating that we welcome collaborations. We welcome, our data are there for everyone to access, right? It's up on the web now. You can download all this information from NCBI. We have part of the value, let's say, of this type of food safety surveillance work that extends across the federal government is get the data out there, get reliable data out there in a timely fashion so that anyone who can act on it has a good scientific foundation for those actions and those decisions. And so in NARMS in a month, hopefully we'll have that data in accessible format for everyone. And then ideally that could lead to questions that we could help answer together. I believe that is my last slide, and I thank you very much for attending today, and I'll turn it back over.
Thank you so much, Dr. McDermott. If there's any questions for Dr. McDermott, please put them in the Q&A box, and I'd like to start with a question. Again, thank you so much for the presentation, and we could really see behind the scenes that truly it is a one health approach, especially with the addition of the environment. But for our colleagues that have joined us from lower middle income countries, Dr. McDermott, with all your experience, what is your advice for them? How do they prioritize, or what is their next step?
Yeah, that's a very good question and a difficult one. I would say, first of all, I mentioned sequencing as a leapfrog technology, and obviously, if I had the wherewithal to do it, and we're starting, let's say, a AMR surveillance system today, there's a lot of caveats about this because you need bandwidth, you need computing power, and you need affordable reagents, and that can be difficult sometimes in low and middle income countries to achieve. But the advantages of sequencing for AMR surveillance are so vast that, if possible, I would do that rather than, because the old system is you have to buy susceptibility testing panels. There's still a lot of outlays involved, susceptibility testing panels, serotyping. I didn't mention that, but you don't have to buy high quality typing in a sera for salmonella surveillance anymore, by and large, because we can predict the serotype for the top several hundred salmonella serotypes just from the genome. So pulse field, if you're looking at strain relationships, you don't need that anymore. That's all inferred in the genome, functional features of the organism, et cetera. So if my advice would be, if you can, to use that technology rather than start with scratch with the old methods that I defined as integrated surveillance. But I know there's a lot of problems with that. Now, that's if the technologies can be implemented, I think that's pretty important. As far as how to design a program, you have to start with the food supply, and you have to start with salmonella, I would say, in terms of foodborne enterics, and you have to start probably with poultry, which is in most cases probably the most consumed product. Maybe even before you get to food, you just start with human clinical isolates, and you build on from there. Now, the WHO Advisory Group on Integrated Surveillance has a sort of stepwise map outlined that would answer that question, and I would refer people to the AGISAR documents.
Thank you. And again, please enter your questions. Another question for you, Dr. McDermott. So given the phenotypic genotyping data that you had described and the genomic cutoff with MIC, do you see, even for developed countries such as ourselves, going straight to using whole genome sequencing for like MIC's clinical practice?
Yeah, that's different. So I would say we're not there yet. As a surveillance tool where, let's say, you're looking at baseline transmission, spread, et cetera, of resistance as a public health activity, yeah, I think you could use genomics alone. If you're looking at the guide therapy in infected individuals, there are some tests out there based on genetics that are approved. There are PCR methods. I think TB has a couple because TB is so hard to grow and et cetera. So there are some methods, genomic-based methods to guide therapy. I don't think whole genome sequencing is quite there yet for most pathogens, but I think in the future it will be.
It's certainly much faster to immediately for many human clinical cases. So we do have a question in the box for you from Dr. Shamila Nagatu. In the sampling for AMR, particularly animal origin, why not include non-food producing animals, but equally exposed to the environment. For example, in our, I'm not sure what case coins. I don't know, Dr. McDermott, if you can see the question box.
I'm sorry, I have to move my camera around here.
I think essentially it's asking equines. Thank you. So so animals equally exposed to the environment, for example, equines.
Yeah, so good question. I mean, part of it is, well, I'll speak from the FDA point of view. You know, there has to be some hazard, identifiable hazard that impacts human health. Probably would be the first question. The second one would be animal health, and you know, it's a question of resources, right? It's the same for us as anyone else. The goal is to deploy resources across all the variable pieces of the program in a way that is, so let's say, the best use of those resources. I would like to know some of those questions too, but it's really resource dependent how deep and wide you can go in terms of the type of testing. But it's a fair question. One we grapple with constantly is, are we doing too much testing over here? Should we move resources so that we can look in these new niches and get better information and get better science? So yeah, it's always a balancing act.
Great. Additional question from Dr. Tom Whittam, whom you know. Thank you, Dr. McDermott, for the talk. Briefly about how NARM surveillance data is used to inform prevention activities. So how is NARMS used for prevention activities? We could discuss that.
Yeah, prevention of AMR, I presume. I mean, I think, from a policy, let's say regulatory standpoint, I mean, NARMS has been used a couple of times, once to withdraw the approval of fluoroquinolones for poultry. So, that was based on Campylobacter data. But, one thing it did, it prevented salmonella resistance to quinolones from emerging as well. And if you look at the U.S. data compared to Europe, we have very, very far fewer resistance problems in the food supply. And one of the most obvious ones is we have no fluoroquinolone resistance in poultry until Infantis blew in the window, if you will. So I guess in that sense, it's a preventive practice in terms of how you regulate the label and how it can be used. I'm not sure if that's what you're getting at, Tom, but it's also when the FDA put out a guidance for that limited extended spectrum beta lactamases, and some of them in some food animals. It was partly based on NARMS. It goes into our risk assessment models when new antimicrobials are approved at FDA, and that might put restrictions on use, right? Like restrict the label, how it can be used, et cetera, to try to avoid resistance from developing from a new approval. Maybe that answers your question.
So, yeah, I think two more questions and then we'll finish off the session. So is there any advantage for overlapping transcriptome data to the genomic data to improve the predictive power of antibiotic resistance?
That's a good, I was surprised to see how well the correlations were between just looking for known resistance genes and MICs at the clinical breakpoint. So if that's your goal, I'd say, no, you don't need it for that. You don't need it. It probably wouldn't close that many data gaps where there's discordance between phenotype and genotype. In fact, most of what we've seen, and the Europeans have confirmed this, the Danes, is usually it's bad susceptibility testing that accounts for the discrepancies. So that might be more important when we start looking at the use of genomics for guiding clinical therapy, where you'd want to see those expression levels, because you do see wild type coding regions that aren't expressed sometimes, in which case you might, in a desperate situation, be able to use a drug even though a silent gene is present. So it might be more relevant to that part of it.
Great. So then the last question for the session from Dr. Paul Plummer. Traditionally, NARMS has struggled in sustainable funding at times. As NARMS look to expand the scope and the effort, how do you see this impacting this? In other words, does the expansion of the effort dilute the current effort?
Thank you, Paul, for that question. Yes, we've had that several times, and watching resources is pretty much my full-time job, so I'm very appreciative of that. First of all, I would say that we haven't sacrificed scope for depth, right? Although we're trying to get to these new sources of information, we're still digging deeper into isolates from animals raised with antibiotics. And I mentioned the addition of seafood and veal and lamb and goats and catfish, and also looking at more bacterial genera. So far, and the other thing that's happened is with the One Health framework, and the need for more disciplines to interact, people have sort of stepped up with in-kind support. So EPA is doing that on the environmental piece. We're getting NOL and VetLearn on the animal pathogen piece. So there's been more in-kind support. But yeah, it's not going to be possible to do, say, probabilistic sampling of national waterways without more resources. So if that doesn't happen, we'll have to rethink how to achieve it. But yeah, it's always resources are always the most important question, I think.
Yep, for U.S. and elsewhere. Thank you again, Dr. McDermott, for this wonderful plenary for the antimicrobial resistance. And again, if there's other questions, please type in the box and we'll try to share it with Dr. McDermott. I'd like to hand the session over to Dr. Tom Whidden, who will be chairing the panel session. Thank you.
Okay, thanks for that, and thanks, Dr. McDermott, for a great presentation. We appreciate you joining the session this morning. And thanks everybody for joining the panel session on AMR. I think we've got a great panel today. So our first presenter will be Dr. Dubra Diaz Campos, who is an assistant professor of clinical microbiology here at The Ohio State University. So Dr. Dubra originally got her DVM from the University of Zulia, Maracaibo, Venezuela. She later did her PhD at Auburn University and a residency at Washington State. She currently runs our clinical microbiology lab at the Ohio State University College of Veterinary Medicine. She's also one of the leaders of our antimicrobial stewardship program and is engaged in teaching and research related to antimicrobial resistance. So Dr. Dubra, I'll let you take it from there.
Dr. Diaz Campos, you're still on mute.
Good morning, good afternoon, everybody. I think you need to mute if you want people to listen to you. So are you able to see my screen now?
Yes, but it is not in full screen mode.
Okay.
Now it is in presenter mode.
Okay. Now it's working.
Yes, perfect. Thank you.
So good morning and good afternoon, everybody. It's really a pleasure to be here today, and I really appreciate the invitation from the organizers of this initiative, because not all the time veterinary microbiologists have the time to